But great.
I think.
Okay. Oops. Your camera's like right here, so.
Yeah, that's not being. That's fine. They would do something. And this student is. There's a lot of. And also, okay.
No, you should take that for yours. This is fine. I mean, this is like, yeah, if there are people like standing up in the back, I'm a bit more annoyed. It is a little like humid in here though. It's a little buggy. It'll be fine. I'll just start sweating more.
It's on.
It's on what?
It's on me.
Yeah, it's on seat. I see. Yeah, yeah.
You know, I set the temperature so we know what it's like to
Yeah.
really.
I was debating starting off with some like Glass Animals, Heat Wave song, like music video. Want to do it?
All right, I think we'll get started. Thank you folks online, but also in the room. It's my honor today to introduce Drew Villeneuve. Drew has been here for 4 years, comes to us from UMass Amherst and then Bowdoin College before that. Drew actually tracked me down before I even had a job here. I was in Vermont, this was during COVID, and we had a grant that we were awarded and it was posted on the NSF website. And like the next day, I get this email from like a random prospective grad student. Mind you, I don't have a job yet. And it's like wants to do a PhD. And so that was back in, I guess, 2020. Like 2021.
Well, I think it's 2019.
2019.
Yeah.
Yeah. So I think it's for me, it's been a joy to have Drew in the lab. He, I think, epitomizes what it means to do a PhD, which I think is a lot of stumbling around a dark room for a while, going down lots of different forked paths and kind of finding your way, and then coming up with a really cool project that is truly his own. And then really excited that in just a few months, he'll be headed off to Friday Harbor Labs at the University of Washington for a postdoc. So thanks, Drew. Take it away.
Thank you, Easton. Awesome. Yeah, thanks, Easton. Thanks everyone for being here. It's just really huge privilege to, like, after 4 years of really hard work, be able to share everything that I've been doing. So this is just really exciting. Thank you all for being here. I think you're all very lucky that this is capped at 1 hour because we could go all day about all sorts of fun things. But in my PhD, I used ecophysiological models to try to predict the responses of ectotherms or cold-blooded organisms to marine heat waves. So first, I want to start with a little audience participation. This may be going to be for people in the room. I took this photo in the intertidal off the coast of Spain a couple of years ago, and it's just really lovely. I want you to imagine that you are a crab or a mussel or whatever your favorite marine critter is. And I want you to think, what are the environmental challenges that you might face in this picture?
Hot.
Okay, it looks hot. Yeah, I did say it was the southern coast of Spain. That's actually Africa and Morocco in the background, which is pretty cool. Right, so it looks like it's pretty hot. Competition, yeah, we see all sorts of different turf and algal species kind of duking it out there in this really nice tide pool. Yeah. So I also want you to kind of think now, well, are those constant or variable stressors, right? And I think it's easy when we go to sites like this that we kind of get a snapshot of what's happening. But in reality, like biology and ecology are so dynamic, so time integrated, and it's really difficult, I think, to be able to have a really good picture of the variation through time that a lot of like the stressors in a lot of these ecosystems. And this is really important because we're trying to be able to predict the populations of marine organisms under a more variable and warmer future. So as we've all probably seen, a similar figure to this, we're seeing the mean global increase in mean temperatures under different emissions scenarios. I wish I could get rid of that stop sharing thing at the top, but and you can see that it's this gradual increase over time. You can think about this as a push or the up escalator of climate warming, climate forcing. But we know this is not a realistic picture of what actually temperature on the ground for organisms looks like. There are these pulses. You can just say that's weather. Whether the temperature varies at a much finer time scale than kind of these climactic predictions and models speculate. And this is a little obvious, I think, to a lot of us. You can also think about this as like a bouncing ball, where it's kind of the variation or the range of ball bounces don't change over time. But there's that variation in how this weather can kind of oscillate. But one of the issues with climate change is that on top of this escalator of climate change, we have the bouncing ball of weather and climate variability. And so even though the variability, the weather is not becoming more necessarily extreme by itself, the baseline is increasing. So this variability is really important too, because it's at a fine time scale that is actually the time scale of which many organisms are operating. So think about the pace of life for many organisms. As an example, this is a great figure from a great paper by Matt Sasaki. We're seeing a silver slide, which can live multiple years. It's experiencing seasonal variation. But if we kind of zoom in and we look at like copepods or phytoplankton, they might live a couple of weeks. So a lot of the question is, how each of these organisms experiences temperature variability and climate, long-term climate changes can be very different depending on their biological adaptations. So when we talk about extreme weather in the oceans, we're usually talking about marine heat waves, and this is a kind of a statistical climatological concept that's based on extreme value theory. And essentially, it's saying that if we have a long-term record of temperature, say 30 years, we can look at some threshold, the 90th percentile, and look to see when temperatures might exceed that long-term threshold. And so this tells us what might be extreme for a given location. And on this plot, it's a little hard to see, but there's a dotted red line on top of that gray ribbon. That's the 90th percentile. So anytime that kind of multicolored spaghetti, which is the each year's annual temperature, exceeds that, that's a heat wave. We can also categorize the extremity. So in a very similar way that we can give categories like hurricanes or different categories of other extreme events, we can apply this kind of statistical framework to say how extreme a heat wave is based on how extreme in temperature it is. So this is kind of like the baseline for how a lot of the oceanographic literature understands marine heat waves. And not only are this like temporal phenomenon, but they're also a spatial phenomenon. And this is just showing a map of like global heat waves over time. And over like the past 2 decades, we've just really seen more and more of these really kind of the rise of these extreme heat waves that are covering more and more of the ocean surface. But the real question is, does this really tell us what is extreme for an organism? Because what you'll also notice is that we have heat waves that are in the tropics, and we also have heat waves in like the Gulf of Alaska, all the way up here. So are those heat waves the same? And we probably should interrogate the organisms themselves. And one of the kind of really obvious knock-on effects that have seen with heat waves have been these mass mortality events. So this is a map just showing some of the recent mass mortality events in the past few decades, really covering the globe. In particular, I want to call attention to these habitat-forming species or foundation species. So these are things like kelp, seagrass meadows, coral reefs, bivalves, et cetera. So these are really important species because they provide 3-dimensional habitat. So like nursery grounds, they also sequester carbon, and they also provide coastal protection. So these are just awesome class of organisms. They're very sensitive to heat stress because they can't move. They can't behaviorally thermoregulate like a fish or a marine mammal or bird would be able to. So they're kind of stuck where they are. And just as some examples of some like really severe events, off the Great Southern Reef in Australia, huge kelp die-off in 2021 in the Pacific Northwest. There's a huge heat dome, which killed literally billions of intertidal organisms. And in the Mediterranean, there's these recurring heat waves that have really impacted a lot of the coral species there. So given that these are really important species for like so many different ecological and biological services, it'd be really great if we could forecast specifically when marine heat wave conditions might cause mass mortality events, and to be able to quantify the damage resulting from these events. And one of the reasons is that we want to be able to provide monitoring tools, so for management and policy interventions that are specifically targeted for different species or different ecosystems. So how do we do this? Well, it's challenging because, and just as like a little vignette, this is philosopher Thomas Nagel, who wrote this little treatise where he asks, what is it like to be a bat? And he says in this that it's impossible for us to know what it's like to be a bat because we can't echolocate. That's like the one sentence thesis. But it's true, we don't have the same like physiological makeup. And so we're bringing our human perspective to try to understand what it's like to be a bat. So what we need to do is we need to figure out what is it like to be a really hot invertebrate. And instead of just saying it's impossible because we're not an oyster, we can actually use different physiological and modeling tools. So in my PhD, across the 3 chapters that I'm going to present you today, I worked with kind of as a core these physiological methods called thermal death time curves. And essentially, when we talk about thermal tolerance, which is kind of delineating heat, sublethal heat stress and lethal heat stress, it's actually really important that we consider the duration of that thermal tolerance. So think about temperature as a dose that's dependent on time. And so what we see in this figure here, this is real data that's showing the collapse time of an organism, of an oyster, as temperature increases. What you'll notice is that as temperature becomes higher, that resistance time, the time that this oyster can withstand that temperature, is decreasing in a nonlinear fashion. We call this and this is, negative log linear pattern. And what's really cool is that we can log transform that y-axis and we get this really nice tractable linear model, which is really nice to work with. What's really cool about these thermal death time curves is that for ectotherms, cold-blooded organisms, this linear relationship is really highly conserved. So we see this kind of shape, but what is different is the slope and intercept. So you can have a line, but it can vary kind of in the actual like position on your kind of mapping surface. And so what this kind of formula tells us is that for each degree of warming, survival is going to decrease by some factor of 10, which also has to do with these 2 parameters, CT max, which I'm going to refer to here. That's the intercept. You can think about that as the acute tolerance. Z, that is the has relation to the slope of this line, and it kind of gives you a scaling factor of tolerance. So we can conduct laboratory experiments to build these thermal death time curves in the lab. But kind of the next step is that we can build thermal tolerance landscapes. So in this talk, might see TDTs. I'm referring to kind of the experimental curve. TTLs refers to this kind of next step where we create a continuous response surface of survival across different timescales. So in this kind of sample image, we have a bunch of different organisms across 3 temperatures that have different times to death. And then we can kind of take these slices of survival probability that are at this kind of negative angle in those dotted lines, and we can interpolate out to create this response landscape. And this is, again, the response surface cousin of a TDT curve. And it gives us, for any combination of temperature exposure and a length of time, gives us a survival probability. So it's a probabilistic landscape. And kind of the last step of why these are really powerful and I ended up working with these is that these TTLs, or thermal tolerance landscapes, encode continuous survival rates. So basically, for any combination of time and temperature exposure you can think of, we can derive what the survival probability is. So through kind of a set of this modeling framework, we can We can combine the thermal tolerance landscape that we've built from laboratory experiments. We can also get the field or the body temperature of an organism. It could be in the field, could also be in the lab. And then we can calculate the injury accumulation over time using this dynamic tolerance model. I don't really have time to go into exactly how it works, so just kind of think about it as a little bit of a black box for now. And this dynamic model takes as an input the thermal tolerance landscape for your species of study and the body temperature. And the output is this survival trajectory through time, which is that red line. So that for a given heat wave or temperature time series of interest, we can calculate the end survival and assess the severity of that potential heat stress. So I became really inspired when I was digging around to figure out what I want to do for my PhD by kind of the potential of this method for integrating time and trying to basically scale up individual performance and physiology across larger spatial and temporal scales. So in chapter 1, I'm going to show you kind of a computer simulation project I did as my first chapter, where I was interested in understanding the marine heat wave interactions with biological and climatological variability. And specifically, I was interested in how do mechanistic physiological methods like the thermal death time curve and thermal tolerance landscapes improve on the climatological marine heat wave concept. In chapter 2, I leave kind of simulation land and I go into a very specific local example where I predicted thermal mortality of intertidal oysters in a New Hampshire estuary. And specifically, I was interested, can we, with a well-parameterized model, predict intertidal mortality at a very fine scale? And then for my last chapter, I'm going to expand that scope a bit, and we're going to take a visit over the Mediterranean to understand the thermal risk landscape of marine heat waves to Gorgonian corals, and specifically trying to understand what constitutes a risky heat wave in terms of how probable a marine heat wave is and what the expected mortality would be. So for chapter 1, as I said, we took a little simulation study. So this is all using synthetic data, and it's a sandbox approach, and to basically allow us to explore different kind of interactions of biological and climatological variability when we're trying to predict thermal stress. So first, I was interested in what are the implications for species with different thermal death time curves. So as I showed you earlier, thermal death time curves have a slope and intercept, but those can vary. And so what's very possible, and actually there's some examples of this, is that you can have in the same ecosystem organisms that have these intersecting curves. So that's kind of the first thing we're going to investigate. And the second is what types of marine heat waves have the largest impact on organism mortality. So in this very extreme example, we have this really short and hot heat spike, and we have this kind of more moderate blue heat wave. So which one is going to have the largest impact on organisms? So to do this, I basically built marine heat waves in synthetic organisms on my laptop. So this here in red is showing a thermal death time curve for this hypothetical bivalve species. I based the parameters behind this model on marine bivalves. So it's definitely grounded in some biological reality. So I could do this for a couple different, bivalves or different hypothetical organisms. And I then simulated like the simplest heat wave you could possibly imagine, which is one that has a magnitude or a maximum kind of temperature that it reaches, and then one that has a duration. So the impact is that you get this like witch's hat looking like temperature series. Obviously, this is not very realistic, right? In the real world, we see so much more variability. But for the purposes of this project, we're just really interested in understanding how the duration and magnitude of temperature stress kind of interact with biological variability. And so, all right, we can combine the heat wave, this blue heat wave time series with the thermal tolerance landscape for this for this synthetic organism, put those together in a dynamic tolerance model. And we can derive at this red line here, the survivorship trajectory over time. So this heat wave yielded a survival of 90%. We can then repeat this entire process, rinse and repeat for 527 different marine heat waves where I vary the magnitude and the duration and basically look at all possible combinations and getting this like response surface. So what you'll see later is that I'll have like on the x-axis will be marine heat wave duration, y-axis will be the magnitude, and it's going to be colored by survival because we've input survival for the corresponding heat wave components. Okay, this is basically creating this bestiary of heat waves because it'd be really hard and maybe not very fun to have to conduct 527 different heat waves in the lab, sacrifice a bunch of organisms. So this is kind of one of the nice things about a simulation sandbox approach. Okay, so the first thing is I wanted to see if there'd be winners or losers between different heat wave types depending on what your thermal death time curve looks like. So we have two here. Here are two synthetic species, a chronic tolerator, and in red, a acute tolerator. And what you'll see is that this vertical dashed line indicates where these curves intersect, which is at 36 degrees. They should die at the exactly same time, 30 hours. What you'll notice is that above temperatures above 36 degrees, this red species, the acute species, is able to persist for longer. So we might say that it has like higher fitness for a given heat event. Concurrently, you might look at, if you look at temperatures below 36 degrees, this blue species is able to persist for longer. So you might say the chronic tolerator is more likely, is going to have more fitness over a heat wave. And this is important because a lot of times when we're conducting thermal tolerance experiments that are only thinking about maybe one duration, which is relatively common in a lot of literature, we might say that this red species performs the best. It is able to survive for longer at 42 degrees. It's going to be able to survive heat waves much better than the blue species. But when we look at the survival response surface of like all the different heat wave simulations we conducted, we'll notice that the red species only performs better in this kind of very narrow sliver because it's only performing better over heat exposures that are less than 30 hours. Meanwhile, this blue species is actually able to tolerate these more chronic, long-term heat waves much better. And this is important because in the oceans, a lot of these heat wave events aren't measured in hours, they're measured in days and weeks. And so this really kind of calls to attention that when we're trying to do these realistic predictions in the ocean, we need to be thinking about this kind of chronic and acute tolerance that different organisms might have. So just maybe remember this intersecting thermal death time curve when we revisit chapters 2 and 3. Okay, the next thing from this chapter that we found was that what I realized is that if you were to look at the survival response surface, so on the x-axis, heat wave duration, y-axis, the magnitude of those heat waves, we could connect heat waves that result in very similar survival with these white lines or these like survival isoclines. And if we take kind of the extreme ends, this blue dot and red dot, those are two very different looking heat waves. We have one that's very short and hot and one that's broader and not as extreme. Based on this model, the survival is actually the exact same. The trajectory through time is a little different because one has this kind of different relationship with the accumulation rate of injury over time, but it should result in the same mortality, which is pretty interesting, I think. And this was kind of the germ of the idea behind my chapter 3, which was trying to understand, well, if they should result in the same amount of mortality, how common are each of those in situ? So which one is actually more risky? So in conclusion from chapter 1, I showed that acute thermal tolerance does not really tell us the whole story, especially when we're trying to measure heat stress and mortality over these like much longer timescales. And that there's also many different heat waves that could result in equivalent mortality. Okay, so in chapter 2, I then wanted to kind of expand into using these methods in a kind of a real world example. So if we accept that these thermal tolerance landscapes correctly describe the mechanisms of thermal stress, we should be able to precisely build a well-parameterized model with well-parameterized thermal death time curves and predict survival. But the question is, in the intertidal zone, is persistence to heat stress different at low and high tide? Is that going to complicate our ability to predict mortality? And so this is what's important to think about too in the intertidal is that we can't necessarily think about just water temperature or just the ambient air temperature. We need to actually focus much more on body temperature. So thinking about what's it like to be a very hot invertebrate, right? So if you're an oyster and I put you in a flask of water, if you are cooler than the water, gradually, or not that gradually, over time, you will match the water temperature, the water temperature with your body temperature. And this is because water is really conductive, high specific heat. We're gonna move on to a goal because I like this figure. Conversely, air temperature is not the only driver of body temperature. So solar radiation, this wind speed, ambient humidity, the material and the conductance of the substrate that an organism is on, all really influence the body temperature, which means that you can have a persistent higher body temperature than air temperature. So we really need to kind of account for body temperature when we're doing these predictions and not just what we're measuring from like meteorological observations. So for this chapter, I used the eastern oyster, Chrysostrea virginica, which is our native oyster here in New Hampshire. And in this chapter, I first conducted thermal death time experiments for oysters that are submerged in water and those that are exposed like they would be at low tide. I then conducted field surveys to measure oyster survival in the field here in New Hampshire, and then also measure body temperature of those oysters. I then wanted to develop a model for predicting intertidal heat stress, and then I wanted to compare my model predictions against the field observations. So the first part of this was conducting laboratory experiments to measure time to death. And so what I used a seawater system at our coastal marine lab here, and air incubators here on campus to expose oysters to various different temperatures. And instead of just seeing how many died at a certain amount of time, we leave those oysters in the water or in this incubator chamber until we detect death. We measure death by measuring cardiac activity with these infrared loggers. So instead of like checking every day or checking every 6 hours, which should be a lot, we are able to use these sensors to basically get the precise time of death based on the cessation of heart rate. So the results from these experiments is that we see this really interesting intersecting thermal death time curve. How cool. So above 37 degrees, we see that oysters that are exposed, like they would be at low tide, are actually able to survive longer than oysters that are submerged below water. So this might be a little surprising. It's like, okay, it's a marine organism. Why is it able to persist better at low tide rather than at high tide at these high temperatures? And it's because a lot of intertidal organisms have evolved this metabolic depression pathway to basically deal with this twice, at least here in New England, we have this twice daily tide. So they have to be adapted to kind of deal with these conditions. So they are able to depress their metabolism, reduce oxygen consumption because they're using anaerobic pathways, and just kind of conserve energy until the tide comes back in. So this is an adaptation that they've developed. There's also this potential for evaporative cooling. I'll just also add, because they have water and they can literally evaporate that water off by gaping their shell, and that also potentially is able to cool them down very slightly compared to ambient air temperature. What's also interesting is that below 37 degrees, the oysters in water persist for longer. And this is because we're starting to talk about exposures of multiple days. So these oysters are beginning to desiccate out a bit. And that can have issues like collapsing gill and like dehydrating gill tissue, which has a lot of impacts on, for example, getting oxygen, which is very important. What else I want to say here? Yeah. So once we had built the thermal death time curves, the next step was to do some monitoring of survival in the field. So at 2 monitoring sites in Great Bay, Great Bay is our inland estuary off the Gulf of Maine. We had these 2 sites, Jackson Estuarine Lab, or GEL, and Brackett's Point. And I deployed at each site 40 oysters, which were co-located with temperature loggers, which were measuring a proxy for body temperature. So not collecting air temperature, but actually measuring, like on these pavers, what it's like when you're baking on basically a piece of cement. And so every 1 to 2 weeks over the summer, kind of depending on whether the weather forecast was telling me it was going to be very hot, we would go out and count the number of surviving oysters. And with this temperature logger, we're able then to also partition out when a temperature observation is at low tide or exposed and when it's underwater. And so here at Brackett's Point, you can see kind of these much larger spikes when the logger is exposed at low tide, It goes up to like 48 degrees Celsius, which is crazy. We're in New England. We're not in like, South Carolina or anything like that. Okay, so the next part of this is I need to build a model to predict mortality in the intertidal. So I decided to break up how stress is accumulated in oysters by focusing on single tidal cycles. So an oyster might start off at the waterline. The water goes up, water comes down, exposes the oysters, and then goes back up to their height. So the idea behind this is that oysters are able to recover a bit of their stress at high tide, and then they accumulate stress at low tide. So the upper part of this panel is showing just the tide height in feet. The middle panel is showing temperature, and you have broken this out in red is when they're exposed at low tide. And that bottom panel is showing the survival trajectory. So you can see that because the temperature wasn't too high, the survival is pretty close to 100%, and that drops off right down to, I think it's like 94, 95%. So 5% of this population died in a single tide cycle. And we've run this with the two thermal death time curves, thermal tolerance landscapes from the experiments that we conducted. And we then rinse and repeat this for every tide cycle and the temperature that we observed and accumulate mortality. Okay, so now we get to compare our model predictions against our field observations. So here we got the temperature at Brackett's Point, and I'm now going to show you the field observations and the model predictions. And 85% of the oysters survived in the field, but our model told us 23% would survive. So that's a pretty big overprediction of mortality. It's not great. Let's look at the other side, gel. So we've got our temperature time series. And we see something kind of more funky where our model begins to initially overpredict mortality, and then there's this big mortality in the field in the middle of July, and then it kind of levels off again. So this is a little weird what happened at gel. It could be temperature. There's also construction that was active at the Jackson lab when we were doing the study, and there could have been construction turbidity, some disease, potentially other factors. And I'm going to actually come back to this in a second. Overall, the model didn't perform that well. This is kind of how science goes sometimes. But one of the kind of hypotheses I had was that when I was building the thermal death time curves and conducting these lab experiments, the oysters I used were acclimating in a water tank with ambient seawater that was like 16 degrees Celsius, so which is matching kind of the ambient water temperature. That's not what the oysters that I put in the field were acclimating to. They were fully exposed to an intertidal time series where at low tide, they were becoming hotter and hotter and hotter as the summer went on and going from sublethal to lethal conditions. So the acclimation didn't really match between our field and our lab experiments. So I could stop there and just be like, oh, well, like that's how science goes. But instead, we could then ask, well, can we make the model predict survival in the field accurately by trying to match the right parameter set of thermal death time curves to the field data? And we do what I've been calling a simulation-based calibration, where we're going to re-simulate the model survival many times and see what matches that field data. So what you do is you build a menu of thermal death time curves for exposed and low tide oysters and oysters that are in water. And so here I'm showing you 9 curves. And because I used 2 curves for my predictions, that gives you 81 different combinations, unique combinations of a parameter set. What that means is that I then predicted 81 different survival trajectories through time, and then I find which data set matches the field data most closely using root mean square error. Okay, so getting to those results, this is going to look a little different than the other figure I showed you. It's showing the % change in survival between monitoring dates. So the % change when I go out on time period 1 and time period 2. In black is the original model prediction. In red is the observed field mortality. All the little gray lines are the candidate predictions that we're interested in. And blue is the best model prediction. What you'll notice is that in all the different, at gel, in all the candidate, actually, before I get to that, let's look at our root mean square error. We were looking for a low number, nice low number at Brackett's Point, slightly higher at Jackson Lab. And this higher number at Jackson is kind of due to this big spike in mortality in the middle of summer. Now, the cool thing is that in looking at all the different combinations of thermal death time curves, we're not able to replicate this spike in mortality in the middle of summer, which leads us to believe that this is actually a non-heat death related mortality event. And that's the cool thing about this model, is that it allows us to actually kind of use it as a null model and see this is the mortality we would expect if temperature is the only stressor. Here, there's clearly something else going on, so we should maybe look into that into the future. And just to show you kind of like the raw data looks like, again, the color symbology is the same. Red is the fuel data, blue is our improved model prediction. You can see Brackett's point fits really nicely, gel fits very nicely, and then you get to that sudden mortality event, and then it kind of levels off again and actually matches that trajectory after that non-heat mortality. So in conclusion, we show that exposed and submerged heat stress are not the same for intertidal oysters. Critically, if we want to build these accurate field predictions of mortality, we need to account for acclimation and kind of these this heat hardening that can and plasticity that these organisms accrue. And there's also this potential for site-specific forecasting. So now that we've got an improved model, we could start to predict mortality at a scale that might be more of interest to managers or oyster farmers and other kind of very specific case studies. Okay, we were focusing on one location, two very closely linked sites at a very small spatial scale. I became interested in, can we then expand this thermal death time framework to predict at a regional, multi-event, multi-year scale? So we're going to kind of take a trip across the Atlantic to the Mediterranean. And in chapter 3, I mapped Mediterranean basin-wide mortality of this Gorgonian coral, Paramurcia clavata. And as a note, I started this project because I was reading some papers about these mass mortality events in the Mediterranean and kind of got linked up with this group of researchers at the University of Barcelona. And they invited me to kind of use some of their data and to try to apply these models to this case study. So much of the field data and laboratory data was collected by that team. This species, it's a red gorgonian. These are heterotrophic, so they are non-photosynthetic. They're really beautiful. They produce these kind of thickets of three-dimensional structure, so really nice for nursery habitat, also like high tourism value for divers. Unfortunately, throughout the Mediterranean has experienced these really severe marine heat wave events in 1999, 2003, 2022, and that's kind of been correlated with these huge mass mortality events in organisms. In this map, you see in 2022, the entire surface area of the Mediterranean experienced a heat wave condition, and that over time, the percentage of the surface area in the Mediterranean experiencing heat waves has reached 100%. So it's a rapidly warming, very stressful area of the ocean. My collaborators at the University of Barcelona have been doing long-term monitoring of populations, and so you can kind of tell or diagnose mortality in these corals by looking at live corals, and then every year they go back and they look for necrosis of tissue. So basically, tissue is just sloughing off after death and leaves this white skeleton. So instead of mortality, we're thinking about the percentage of colonies that are experiencing this kind of tissue death. And when they look at the field data over time, this is at a couple of sites kind of brought together. We can see that the y-axis here is the affected colonies percentage. It's gone upward over time. In 2022 was that really, really hot heat wave year. And you can see that big, that huge percentage of affected colonies. So I became interested, can we use this thermal tolerance landscape, dynamic tolerance model method to actually predict local mortality at specific sites, first to kind of validate the approach, and then expanding to the Western Mediterranean to ask what types of marine heat wave mortality events are the most common, and what are going to be the most risky based on the occurrence probability and the kind of corresponding mortality risk of those corals. So to map mortality of this Gorgonian, the first thing is we used an ecological niche model, which is kind of just the species distribution of this Gorgonian from one of the collaborators we're working with, Laura Figueroa Ferrando. And that's all of these black pixels. So they're hugging the coastline, but they're actually kind of this deeper water species. They tend to not live above 20 meters in depth. And in kind of the rainbow colors is our temperature series that we're using. It's this Copernicus reanalysis temperature product. We have 38 years of temperature. What's really cool is it's not sea surface temperature. It's actually depth banded. So we were able to get water temperature at 20 meters, which is really important because there is a pretty severe thermocline in a lot of areas of the Mediterranean. So we want to get temperature at depth. So I have 38 years of data in all of those black pixels because I basically cropped out all the areas where this coral is not found. And you do that, and you try to, if you identify how many heat waves there is, 120,000 heat waves over the past 38 years in all of those pixels at 20 meters. And also kind of one of the reasons I decided to take up this project is that there's already an existing thermal tolerance landscape data for this species based on another collaborator's PhD research. They weren't necessarily conducting these experiments with like the thermal death time concept in mind, but it's just been really cool to like see how, previously collected data can be used for like these really kind of cool future works. So kind of doing something that is hopefully familiar by now, we combine individual heat waves so 120,000 different time series and the thermal tolerance landscape into the dynamic survival model, and we're able to get per marine heat wave survival. And so again, this is the predicted survival per heat wave. Before I dive into the results of the entire kind of region that I showed you, let's go into a model validation. So does this actually work? Collaborators at the University of Barcelona have been collecting long-term monitoring data at these two sites in the Mediterranean. So one at Medes in Spain and other at Scandola in France. You can just hear another picture of kind of the colony death. And based on kind of the years of monitoring data they have, I then used temperature data from these locations from temperature loggers at each of these two sites, and then compared the modeled and field predictions. So at Medes in Spain, we see, I think, a really nice fit. It's matching pretty closely in some years to the actual field observed mortality, and generally it's showing really well like that trend over time. And then at Scandola in France, it tends to overpredict again, which is an interesting theme. This could be due to a couple of things. It could be some plasticity. What's also likely is that there's some local adaptation in this population. It's slightly warmer, so these populations over time may have been able to adapt a higher thermal tolerance. And remember, we're only using one thermal tolerance landscape for this model. So like in the future, we could always build population-specific tolerance landscape models. But nonetheless, it kind of follows the general trend of the field data pretty well. Okay, so once we kind of establish that this does a good job at specific sites, we then compared all of our data across those 120,000 marine heat waves. And this is what the data looks like. There's 120,000 points. X-axis is duration of heat waves. The y-axis is the magnitude of heat waves, and it's colored by the mortality here in this case. And what's cool is that you'll notice that we're not just talking about like a simple triangular heat wave anymore. These are like real life complex time series of temperature. And despite that, you still see this negative log linear thermal death time curve pattern that's kind of cropping up in the survival, which is really cool. It does tell us that kind of those two components explain a ton of variation. So what we next wanted to do is that you can't quite see 120,000 events here. So we then computed the density of marine heat waves. And so basically, I'm going to show you some kind of mountain peaks of density, and just showing where the most common heat waves are. So this little 3D figure kind of just shows you an example of that. And then once we've calculated the density of marine heat waves, we want to group that out by the extremity of mortality that our model predicts. So what heat waves are the most risky? I've categorized these by the four marine heat wave, or the four mortality categories that are used in a lot of like the monitoring literature for this species. So what you're seeing here, it's there's this kind of blob that's shifted over to low duration heat waves, and those are resulting in a very low like colony and necrosis. But they can go up to like 25 degrees, but they were generally quite short. if we start to look at events, marine heat waves that resulted in more mortality and up to 60%, you'll notice that kind of that density is beginning to shift over onto the x-axis. So it's beginning to be dominated more by the duration of the heat wave. And at the most extreme heat waves, they all generally are above 25 degrees as their maximum temperature, but they can have a wide range of durations. So again, thinking back to chapter 1, where I was talking about there's many different roads to kind of get to the same place, you're seeing that here where these ridges are kind of connecting all the heat waves that could result in a similar event. And just the peak of that, of this most extreme heat wave category tells us what is the riskiest heat wave, because it tells us what is the most common kind of combination of heat waves that result in the most extreme mortality. So at 26 degrees in 12 days, those are kind of the most common extreme mortality or extreme colony death events for this species. Kind of the next point is I'm going to just track for you the path of those peaks through marine heat wave space. So what you're seeing here is each of the little black dots there are the like peak of the mountain, and I'm just grouping them instead of by 4 categories, by 10 categories. So grouping more colony necrosis by 10% from 0 to 100%. The color of the arrows or the vectors tells you what is kind of the dominant component of the marine heat wave driving that. Red arrows indicate a path that's dominated or increasing mortality that's driven by increasingly hotter heat waves. Blue arrows tell you increasing mortality is increasingly driven by longer heat waves. And what you'll notice is that up to 30%, heat waves generally are just becoming hotter to drive increasing mortality. But there's some kind of threshold at some point around 25.5 degrees where heat waves generally don't become hotter, they just become longer. And the most extreme mortality events are mostly driven by heat waves becoming much, much longer. And partially because GIFs are fun, but also because you can see the density plot here. You can see how the peaks, which are like the nodes being connected by this red line, and then the funky little like topographic mountains of density are shifting from that y-axis dominance being dominated by magnitude to one that's being dominated by duration. All right, so this has been like a really fun chapter to work on. One of the things we're going to try to work towards as well is, and also kind of thinking about back to that oyster death chapter, is trying to build these mortality forecasting systems. So this is kind of more of a preview of like what I'm hopeful to work on next. So we kind of have these existing heat stress products. One is called the Coral Reef Watch that NOAA produces, and it just shows general coral heat stress. But it's not intentionally, doesn't intentionally use like biological information. It does use what's called degree heating weeks, which is just accumulating the degrees and the number of weeks above some threshold. So it does account for that time component, but it doesn't think about like the different adaptations that species have and kind of the biological side of that. So what, we get this GIF to play? Oh no. This should be a GIF of mortality over time, but it's a map that's showing the survival in this year. We've got 1998, and this would normally showing you the trajectory through time, and you'll notice that most of those pixels are becoming redder and redder and redder. The hope is that we can use these sort of maps to start identifying persistent mortality hotspots and persistent refugia, which can really help with like designing MPAs, conducting like outplanting. And then what's also really exciting is that the Copernicus temperature product that we used behind a lot of these predictions is also a forecast. We can get a 10-day temperature forecast at 20 meters. So we are hoping to be able to extend this to actually have a real-time forecast based on the physiological model of this species. So in conclusion from this chapter, we should consider marine heat wave probability and the mortality impact when decoding biological risk across these large spatial gradients and space and time gradients. We also see that dynamic tolerance models can predict at larger spatial scales. We're able to generally predict tolerance mortality pretty well at 2 sites. And then also we're able to use it across a large basin. So going from that individual to larger spatial populations. And what's really exciting too is that this shows not only for this chapter, but also for chapter 2 on oysters, remote modeling of population health is possible using these mechanistic models, which can really hopefully help kind of if you're in a resource-strapped management environment, you don't need to necessarily have cover every single area that you want to monitor. You can kind of focus in on areas that you might model as being the most impacted. So when we're thinking about, in conclusion, we're trying to think about what are the responses of organisms under a more extreme and variable environment. There's really two things we really need to think about, which is, again, we need to be measuring temperature at an ecologically relevant scale. And by that, I don't even just mean like body temperature. I also just mean we need to think about temperature in time and start thinking more about the accumulation of stress and recovery and how that kind of incorporates into organism performance. And then concurrently with that, we need to think about how organisms are able to integrate time and kind of persist in these like real life field temperature series. And hopefully from there, we can get an understanding of extremity from an organismal perspective, which basically means we need to think about what it's like to be a really hot invertebrate. Okay, that is a very, I still think, brief rundown of my PhD. We could stay here forever, but kind of for the fun part now, I want to start acknowledging so many people. So first from that last chapter, my collaborators in Spain, Paul, Laura, Joaquim, Yanis, Christina, and Danny, they all have been really kind to like kind of let me bring me into their research and to like collaborate with me and produce this chapter. So many interns have helped my journey along the way. Ella, Josie, and Tini basically helped make chapter 2 a reality. That was a ton of work and truly would not have been possible without them. Huge thanks to my committee as well. Easton, obviously, Brittany, Jen, Brian, and Joey. You guys have been just so wonderful to work with, and I really feel like I'm a better scientist from having worked with you. I also thank my funders, Sea Grant, New Hampshire, SMSOE. I also want to give a huge shout out to the Ecological Forecasting Initiative, which provide a lot of critical support in the community through this dissertation. And now for the more fun photo portion of the acknowledgements. QML, my lab has just been such a wonderful place to work. All of my lab mates, like past and present, you guys have just like really helped support me and helped like debug code and also like hang out at conferences, and just it's been so wonderful to get to know you all, and I'll really miss being able to be around you guys. Yeah, it's just been wonderful, especially since I don't live close to UNH, as many of you know. It's been just really nice to still feel accepted and like really part of the group, even when I'm commuting in from Maine. My advisor, Houston, it's just been so wonderful to be able to like have you as my mentor. I also like I'm gonna keep saying this, like I've really grown as a scientist and as a human being under your tutelage. Okay, it's a little animated out of order, but don't think just because he's a quantitative lab and he works on a computer a lot. This is us in the field on computers. Easton also does field work. Here's him drilling, and I think you were very excited about that, about using the hammer drill. But I think that just shows like Easton is just there for all of his advisees, and like he'll go out in the field and to drill holes in rocks because it is fun. But is also just incredible mentor, incredible scientist. Here on the right, as part of a side project, which was supposed to be a PhD chapter and ended up being a side project, I was able to join Easton in Madagascar. What you don't see on those yellow shirts is that they have our photo on the side, and I probably should have worn it actually today. But I'll just always really appreciate having that really fun shirt from our colleagues there. I also want to, and part of the reason why it's been so wonderful to have you in the lab is that Easton has encouraged me to kind of seek out all these really fantastic opportunities. One of which was helping teach in our workshop in Spain with Faith. And what's just been so wonderful about that, I mean, obviously going to Japan to teach R is fantastic. Cheryl Ames here. It was wonderful to see her again because she was my high school intern supervisor at the Smithsonian. And when I was like 14 or 15, and then to come back like all these years later, and like she's now a professor at Tohoku University and like hosted us. And it was just like such a fun full circle to come to. And so this is also just a call, like there's so many mentors in my life, Cheryl and so many others who have like helped me get to this point, and I couldn't do this without them. My family, Margot, my mom and dad, you guys have been so amazing throughout my life and have supported all of my ventures. Basically being the weird science kid for so long. I don't think they had any doubt that I'd be here today, which is, I guess, encouraging because in a PhD, there's always doubts. But I think they saw the path I was on and encouraged me to keep doing that. And lastly, my wonderful partner in life, Laura. We have been together for 8 years. Every single year, one of us has been in grad school. Myself more. I've been in grad school more than she has. But thank you so much. Yeah, you're just such an important part of my life, and you've encouraged me to follow my dreams. You love critters as much as I do, and I really could not have done this PhD without her, so thank you. And I will finish there. Thank you so much. Awesome. I managed to finish with quite a good amount of time left, but if people have questions online, I think you should have the chat feature enabled. I'm going to start with questions here, though, while that kind of comes in. Yeah. 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 Oh,
How do they differ from what you found?
were you a plant? Yeah. Okay. So that question was for chapter 2, because I had a slide about this and I had to cut it. For chapter 2, the model that performed the best, that predicted the field the best, what did those thermal death time curves look like? Let's see if I can pull it up. They look different. What was interesting, while I try to make this work, was that the submerged thermal death time curves looked the same. So they did not become any different. Okay, I think people should see that. Let me get the online people looped in. Okay, there we go. Okay, it's a little small, but I'll start with the part that's below. When I did the simulation, the submerged thermal death time curves were not any different, which is super interesting. It was the exposed thermal death time curves that were at higher tolerance. And what's interesting is that Brackett's Point was the hotter overall site, and that had the like more thermal tolerant curve than gel. So what's interesting is that the thermal death time curves that matched both, that best matched the sites, were slightly different, but only the kind of air exposed thermal death time curves, which definitely tells that if we accept those at face value, we probably can say that there's the acclimation maybe at brackets being warmer actually led to more heat hardening and a higher thermal tolerance curve. Cool. Faith.
Great talk. Can you do chapter 2? And like the idea of like the thermocline that you mentioned in chapter 3.
Yeah.
We talked about the immersed oysters.
Yeah.
What depth were they at? And was there a thermocline in play there?
That is a very good question. We located these oysters high up in intertidal, kind of at the upper range. So when they were submerged, it, depending on the tidal cycle, was probably 0.5 meter at most. In terms of the thermocline, there's probably not much of one. The temperature loggers would have picked up though any kind of change. And so that's the nice thing, is that like whatever, because they were located centimeters apart, whatever the oysters were experiencing, the temperature logger should be picking up. But definitely in Great Bay, there's a lot of mixing, so I'm not sure how much of a thermocline there is. I think there's probably a dissolved oxygen thermocline though, for sure.
Good.
Oh, go ahead.
see, it's what I was thinking is like oyster farms, you know, how they're like a little deeper.
Yeah.
Like that could play a role in like mortality trends.
Yeah, that's the lab has kind of been looking to that a little bit. I think temperature is much less likely to play a role. Like as I wasn't able to show you, but basically when the oysters were submerged, very, very, very little mortality accrued. It was basically all at low tide. So yeah, it's probably more dissolved oxygen and disease at that point.
So I was also wondering about chapter 2, but I was curious how you might expect the thermal death curves to differ between your experimental setup and then oysters that are in like a reef system.
Yeah. Yeah. So the oysters I used were from an oyster farm. So they were cultured. I wanted to use wild oysters, but at least in New Hampshire, they're like all glued to rocks, and you would basically have to use a jackhammer to chisel them off. So we kind of went a more practical route. In the intertidal, if you were to take like here in New England, we do have those intertidal oysters. They tend to be like found under rockweed. In the southern states, they are intertidal and they are like baking in the sun. And what's really cool about them is that they actually kind of all become very narrow and vertical, partially because of crowding, but also potentially to kind of try to escape some of that heat. I think it would really depend on like what type of oyster reefs we're talking about. Yeah. Okay.
It's so fun to see the narrative together. And I'm also really personally excited about how there's a narrative takeaway from both chapters 2 and 3 about the role of adaptation and acclimation that we can draw from your dissertation. I'm wondering, first of all, if you expected those kinds of conclusions. And second of all, like, where do you think we go as a field of spatial modelers and predictive modelers from here? Is there, like, do you think there's a future where we are modeling a surface of potential adaptation and that's a parameter in models? Or do you think that, like, this is something we need to deal with in other ways?
Yeah. So I should, sorry, people online have been hearing these questions. So that question was, did I expect to see adaptation when I was conducting these experiments? And then also kind of what I think the future of like trying to build these better spatial models that might be encountering all of that. I think that's kind of the gist. So I did not initially, like when I was designing these experiments, did not intend to like capture potential acclimation. And so like one of the things was like the results from chapter 2, initially they weren't matching. And like that was a moment of like, oh gosh, the acclimation was like this makes sense. Like even though the oysters were put in the field, were kept at the same, like initially they were kept in a lab at the same temperature in water, but then put out in the field, and that they were able to acclimate to that point. So I did not necessarily design the experiments to capture that, but like that is definitely, I think, like one of the next steps. I think that thinking about plasticity, so thinking about kind of that sublethal priming that can happen. And then I also think being able to account for recovery, because depending on what, after a heat event, what they kind of go back down to before the next heat wave is going to really determine like how much recovery can actually happen. It'd be great for us to do experiments that could, to build models that could kind of encompass all of that. We start to get into, and this is the issue, like any mechanistic model, is that they're really, really data hungry. Like you got to do all sorts of experiments or kind of different like calibration Bayesian methods to identify those parameters that fit. So at some point, it probably is only tractable for like study species or model species. I think it's going to kind of depend, like if we decide in a given system, is it important? And knowing like, oh, if we incorporate this submodel, does that add, how much fidelity does that add to our predictions?
Lots of good comments online or questions online. Mostly just saying congrats, but also someone was asking around recovery.
Okay.
How you handle recovery, where in a lot of terrestrial systems, you would think about recovery maybe at nighttime.
Right.
Temperature. So how do you think about that for what you would do next?
Yeah. So for chapter 2, recovery, we assumed that recovery would happen at low tide, and we assumed that there was at high tide. There's complete recovery that works in New Hampshire because our water temperatures are pretty much completely sublethal. Like they're actually very optimal for oyster growth in many ways. That's why we have oyster aquaculture here. If we wanted to predict like mortality in, say, Georgia, we might have to revisit that assumption that being submerged at high tide yields 100% recovery. And there's some, and again, this is like very new. The couple other teams working in this sphere have like just started to come out with papers that are talking about recovery. And there's like kind of this experimental method you can do to like get at that. But I'm hoping that like in chapter 2, we're really showing that you can adjust these tolerance models to try to emulate what it's like ecologically. What was the second part of that question?
That's great. Yeah, yeah.
Okay.
And we will leave it there for time.
Yeah. Please email me if you have more questions.
Thanks, Drew.
Thanks, guys.
We'll hang out for a minute, but then he's gonna go to his close the word questioning session, 10 to 15 minutes. So.